This is Kanika Gupta, your TA for the session. And as we always do, we'll first have a recap of what you guys must have seen in the videos in the last week, and then we'll take up the questions, the assignment questions, and also some of the questions that I have brought for you guys. Okay, so let's start with the session. Okay. So in the last week, videos that you have seen, that was mostly the continuation of what we were discussing the previous week also, the differential gene expression. That is what we have discussed earlier in the earlier week also. That what can be the levels at which the gene expression can be controlled and can be different in different tissues in a single organism. So that is what a differential gene expression is. That means the regulation of gene expression, single gene, but at different time levels, or we can say at different levels. So that is what we have seen in the last week, and still it is being continued here in this week also. Uh, okay. So what we did saw was what we are continuing here is we have discussed already the differential gene transcription. Now here is the selective nuclear RNA processing. Because if a whole gene makes the full RNA, then depending upon the what is the further processing of the RNA, different type of proteins can be formed, and those different type of proteins can give different kind of expressions, can influence that particular tissue, that particular cell in different ways. That is why this level also determines a differential gene expression. So let's see how does this happens. So if you see here, this is basically a cell in which a RNA is formed from a gene. But if you can see here, we have 1, 2, 3, 4, and 5 exons here, and 1, 2, 3, and 4 introns here. So basically, depending upon which of the exons come together to make the mature mRNA, it will form different kind of proteins, and those different kind of proteins will lead to different kind of expressions or uh, different kind of phenotype in that particular cell and tissue so far. So basically, this is basically the RNA selection. Why RNA selection? Because of the different type of exons that are included in the mature mRNA. Okay, so these are the exons 1, 2, 1, 2, 4, and 5 in this particular mRNA. And for this mRNA, they have chosen 1, 3, and 5, fifth exon. So these 3 exons are making this mRNA, and these 4 exons are making this mRNA, making it a different kind of a protein. Okay, so this was also shown here, like this was an example in which a whole RNA was formed from the gene, but mature RNA, because the protein was ectodermal in expression. So if you can see the introns and exons, so exons in ectoderm is higher comparatively than the endoderm and mesoderm, and introns are almost equal. That means that the particular protein, the exons that were present in that RNA, were having more of an ectodermic expression than endodermic and mesodermic. So lesser exons were included in the endodermic or mesodermal RNA compared to the ones which were going to the ectoderm, if you can see it from the western blot that they have shown it here. Okay, so another example is the cassette exon here, and mutually exclusive exons. So this is an example of type 2 pro-collagen and FGFRF2. This is some kind of a transcription factor. So if you can see again, there are 3 exons here, but only 2 are included in 2B mature chondrocytes, and in the precursor chondrocytes all of the 3 were included. So basically, depending upon which type of a cell is there, what is the stage of the cell, different mRNAs, different exons are included in the mature mRNA, that will lead to the formation of particular protein. So these are both chondrocytes, but this is a precursor and this is the mature chondrocyte. And same goes for FGFR2, the limb ectoderm and the limb mesoderm. So in ectoderm, 7, 8, and 10th exons were included, while in case of the limb mesoderm, 7, 9th, and 10th exons were included from the same RNA or same gene. ठीक है, तो mature RNA, what will be the composition of mature RNA will lead to the differential gene expression in that particular cell type or in that particular tissue. Okay, another example is the alternative 5' splice site. For example, this BCLX2, so this BCL factor, as you must have understood in the video also, is a major factor controlling the apoptosis, the programmed cell death. So what exons are included? Either it is forming BCLXL or BCLXS, can lead to different fates of a particular cell. Either it will go undergo apoptosis or it will be prevented from apoptosis. So this is a phenomenon majorly used in cancer cells also. Cancer cells also showed this kind of a phenomenon, depending upon either they have increased the expression of BCLXL or BCLXS. And the alternative 3' splice site that can be shown in the cordon protein. So again, either it will form a truncated variant 3 or a wild type protein, depending upon the 3' splicing in the RNA, will tell us either a mutant protein is being formed or a wild type protein is being formed. That will lead to again different kind of diseases. It will influence the particular disease in that organism in which this kind of a differentiation happens. So these differential RNA splicings are also they're related to different kind of diseases that occur in some kind of in some organisms. Okay, so this is an alternative splicing of an mRNA in different types of muscles and hepatoma or brain also. So basically, if you can see, this is a whole long complicated RNA, and this RNA, depending upon the different splicings that is being involved, because 5' UTR and general 3' UTR will remain same, but obviously the exons that will be included in that in different tissues will be different. For example, in striated muscle it is different, in striated muscle too it is different. So in striated muscle also it will be differentiated depending upon the location, depending upon the stage of maturation, and many factors that are there. Then in case of smooth muscles also, the exons that will form the mature RNA is different. In myoblast also, myoblasts are basically the muscle cell, so it is different. In non-muscle fibroblasts, it is again a different exons that are included. So again, a single mRNA can lead to different kind of expressions in different tissues, can form different proteins in different tissues depending upon the splicing of that particular RNA, which will form the mature RNA, and that mature RNA will then be responsible for different kind of expressions in that particular tissue or cell. Okay. Next is are the DSCAM gene of Drosophila that can produce 38,016 different types of proteins by alternative nRNA splicing. Okay, so this DSCAM gene of Drosophila can produce this much. Single RNA can produce 38,000 different types of proteins by alternative nRNA splicing. Okay, so basically, if you can see in exon 4 there are 12 alternatives, in exon 6 48 alternatives, in exon 9 it is 33 alternatives, and in exon 17 they have 2 alternatives. So if you do a permutation combination of these different kind of exons, you can have 38,016 different types of proteins or expression from this single RNA. from this single RNA, it can lead to formation of these different types of the proteins. So again, this is a very good example of differential RNA splicing, depending upon basically the splicing of the RNA. So this is an example of this DSCAM protein in which it is specifically required to keep dendrites from the same neuron from adhering to each other, so that there won't be any kind of a crosstalk, or there won't be any kind of a problem in sending the signal further. uh, Crosstalk as in the same neuron. It is not like between two neurons. So DSCAM dependent, if DSCAM protein is coating at the nerve endings, so it will sorry, on the dendrites, then it will lead to what? It will lead to some kind of a repulsion. And there are different, if the DSCAM proteins are not matching, then there will be no repulsion and there will be a crosstalk in between the neurons. So this is basically a wild type kind of a neurons, and these are the ones in which DSCAM is absent. They lack DSCAM. Uh, so this was one of the examples of RNA splicing, of differential RNA splicing. Next, again, this is an example of termination codon within intron. So if again the problem is there in this, these are the disease related examples of RNA splicing. So in wild type, what happens is the termination codon is present in intron, and in mature mRNA basically the introns are spliced out, so there are only exons present. So that can lead to a wild type protein, a functional protein. But if there is some kind of a mutation in which the introns are also included in the mature mRNA, so if you can see it here, the termination codon is present in intron. So before forming a complete mature protein, the translation will be stopped here at the termination codon, and a truncated protein will be formed. That will be a non-functional protein, and that can lead to diseases. For example, here it is muscle hypertrophy. So there can be many kind of a disease related to the RNA splicing also. So it is very important phenomena for formation of a mature RNA from a non-mature RNA, and obviously the alternative splicing, the 3' splicing, the 5' splicing, everything. These are all very important phenomena for a for the formation of a proper functioning protein, which can lead to different fates of a cell or a tissue. Next thing is in the differential gene expression is the selective messenger RNA translation, in which the RNA, which mRNA will be translated, and how it will be translated, what will be the condition when it will be translated. These are also different levels at which the gene expression is controlled or differential gene expression is maintained. So here is the example. In case of no prolactin, the mRNA is degraded quickly, and in case of presence of prolactin, the mRNA will be the half life of mRNA will be increased till 48 hours. So here this mRNA was controlled because of the presence or absence of the prolactin protein. This is the circularized mRNA. So basically, the circularized mRNA is blocked, is masked by different proteins. The translation factors does not bind here to activate this kind of mRNA to lead to the formation of a mature protein, of a functional protein. But when a particular transcription factors comes here, or this CPEB translation factor is phosphorylated, then this maskin, that is making a circular loop kind of a structure on this mRNA, will move from here. So now this mRNA is open for transcription factor to bind, for different polymerases to bind and start the translation, and make the proper functional protein from this mRNA. So this is again a phenomena at on which the translation of mRNA is controlled for differential gene expression. So this is an example from Drosophila, in which bicoid is there to control this, to make this circularized type of expression of inhibition on a mRNA. And when particular transcription factors important for this to are present here, they bind here and they will remove this bicoid from here, so that now it will be active. It will be present for different polymerases and transcription factors to bind here and start the translation for which this mRNA was formed. So this is another level of maintaining the expression for controlling the expression of particular protein or gene. So these are some of the examples in which the mRNAs are stored in oocyte cytoplasm and translated at or near fertilization, and these are controlled by these type of expression, the circularized mRNA, presence or absence of protein. So there are many methods by which these mRNA or formation of these proteins will be controlled. Further, those are the only examples that we were talking about. This is an example of lin 14 mRNA from C. elegans. Again, it is the formation of this loop can be inhibitory sometimes for the mRNA, which leads to stop the translation of that particular mRNA, and these loops will be further then move forward whenever a particular chunk of the mRNA needs to be translated. So this kind of a phenomena initially was seen in C. elegans. Now it is present in many organisms, as you guys must have listened in the lecture also. So miRNA we have discussed in the last class also is the microRNA. So these microRNAs control the expression of a particular gene by binding either a microRNA, or it can be by recruiting some kind of a polymerase there, some kind of an enzyme there which will not let that RNA to get translated. So at many levels miRNA works. So how miRNA actually forms and functions is being shown here. The drosha protein, the dicer proteins are those proteins that can lead to the formation of a mature miRNA, and then that miRNA actually binds to that particular RNA where it needs to repress the translation, and it will stop the translation of that particular mRNA. Sometimes it can recruit this RISC complex, which is for the cleavage of a particular mRNA to stop its expression, to stop the translation of that mRNA. So miRNA works both ways. For an example, in this lymphoid precursor cells, if MIR181 RNA is low, so it will form B cell and it will form T cell at the basal level. But if it has an activated, just a moment, class. Hello? Yes. Yes, sir. Actually, sir, my I don't know what is the problem with Google Meet. It doesn't shows me notification whenever someone wanted to enter. That's why I couldn't admit you. But I've admitted you. Now you can see the session, sir. Yeah. So sorry, class, sorry for the interruption. Yes. So as I was telling you, in these lymphoid precursor cells, if this activated MIR181 mRNA is included, then in that case, it will lead to the formation. It will lead to the formation of enhanced number of B cells and decreased in the number of T cells. So in absence of this, the basal level of B cell and T cell were being formed, but in presence of this, the number of T cells is reduced and the number of B cells is enhanced. So it is a kind of a repressor miRNA basically, because it is a microRNA and it has the function of repression only. So this is an example of how miRNA works. Okay. Okay, so this is the miRNA complex. They are showing how it is activated, how it basically functions in repression of a particular mRNA. So it can either get attached. It can either get attached at the 3' untranslated region, it can recruit some kind of an endonucleus which can digest the poly A tail, which further leads to the either destabilization or reduced expression of that particular mRNA, or it can cause the proteolysis also sometimes by again recruiting some kind of an enzymes on mRNA. So miRNA can actually control the expression of a particular mRNA or a particular protein at all the three levels. Okay, so these are the mRNAs. These are basically telling you that how the localization of a particular mRNA also can affect the expression. So this is the nanos RNA, and these are actually important for the anterior kind of an expression. In posterior part, they are generally sorry, in the posterior part of expression. In anterior they are generally repressed. In posterior only they show the expression, so they generally diffuse to that part and can show the expression there only. In other parts it is inhibited. So this is just to show ki how localization of mRNA also is differentially expressed, and how in same cell, how where they are localized actually matters for their expression. This all you will study when we'll study in detail about the drosophila, how in drosophila differential gene expression works. You will see each and every detail about nanos, about bicoid, about every kind of these genes you will study in drosophila. So again, this is as I told you in the earlier example, the localized protection by whether the mRNA is present in the anterior portion or posterior portion. So if the expression is important for that region, then only that mRNA will be expressed. Otherwise, the expression will be protected by some other proteins that are present there. So here it is HSP83, which prevents the expression of this deadenylase complex, which prevents the expression of this HSP83 mRNA. So it is expressed in case of the posterior part, but it is generally inhibited in case of the anterior. Sorry, it is expressed in anterior, but it is inhibited in posterior. So these are some of the mRNAs which are active when they are being transported. So this is the bicoid mRNA, and this is the Oscar mRNA. They are transported by the motor proteins, the dyneins and the kinesins. So these are the mRNAs which are active when being transported, and obviously wherever their expression is required, they are transported there and get expressed there. Next, this was all about the differential gene expression. Now we come to the genetics in case of in molecular biology and developmental biology everywhere, because this is what the general genetics is. So the father of the genetics is called Gregor Mendel. It is the picture of that famous scientist only. So Gregor Mendel started his experiments on sweet pea flowers. He chose them because obviously maintaining them was easier, and it is a bisexual flower, so it has both the male and the female part. And obviously the generation time, the generations that it needed to study the expression of a particular gene, and to study a particular expression was lesser. So yes, that is why he used sweet pea flowers for his study. So how did he do that? He removed stamens from purple flower. The stamens are the basically male part of a flower, and ovaries are the female part of the flower. So he removed stamens from the purple flowers which were present on the white flowers, and transferred pollens from stamens of white flowers to the carpel of purple flowers. So carpel is basically the female part of this plant, and this carpel has a stigma which receives this pollen, and then after the whole process of fertilization and everything starts. So he removed the stamen from purple flowers and kept the carpel part on the purple flower, and used the stamens of this white flower to transfer the pollen to purple flower, so that it can see what kind of changes is seen in the next generation, in the F1 generation, the first generation. So he pollinated the carpel, pollinated carpel, then matured into the pod. Then he planted those seeds again and then examined the offsprings, the color of the flowers, and the characters that he chose. So parent generation, purple flower was the female part, white flower was the male part. So all the plants in the F1 generation were purple. Then in the next generation, what he saw was 3:1 ratio. So Mendel just checked these heritable characters for 3 generation to see if there is any kind of a variation and what could be the possible reason for these kind of variations in the plants that he has chosen. So he uh, then further saw the results, and according to the results, what would be the case is the dominant trait was the purple and the recessive trait was the white color of the flower. And the flower position, dominant trait was axial, the recessive trait was terminal flowers. Seed color yellow was dominant, green was recessive. Seed shape round was dominant, wrinkled was recessive. That means that whenever the homozygous condition will be there for recessive, then only that trait will be expressed. Otherwise, if a heterozygous condition is there, the dominant trait will always be expressed. In case of Mendel, when he examined these characters, pod shape also inflated was dominant, and this constricted was recessive. Pod color green was dominant, yellow was recessive, and stem length also tall was dominant and dwarf was recessive. And that is how could he say these kind of characters, which character is dominant and which is recessive, because he observed these characters till F2 generation. That means 3 generations he actually observed these characters, and depending upon that he derived some ratios, and he could see that some characters are dominant and some characters are recessive. So how did the results came off in purple flower? First of all, what we think is that in purple flower there can be two conditions possible: either it is an homozygous dominant, either it is an heterozygous dominant. But in case of white flower, it has to be homozygous only, then only the white color will be expressed. Otherwise, if it is a heterozygous condition, the purple color will be expressed. So for initials, he thought that let's see if the purple flowers were homozygous dominant and white flowers were homozygous recessive. And in F1 generation, he got all the purple flowers. This is only possible when the one parent is homozygous dominant. So the heterolygous condition is there. Here we have a capital P, a small p. That means, and all the flowers were purple. That means one character is dominant over other. Then next he did is, in the next generation, he again crossed these flowers in between themselves, and he found that three of the four were dominant and one was recessive and one gave the white flower. That means the ratio came out to be 3:1. This was the case of the complete dominant or recessive kind of a situation. So this was a Punnett square method through which he devised that, and he found that three of the four plants were purple and one was white. This was the general case. This is how a complete dominant or complete recessive character works. Next is the P then again. So this told us about the Mendel's law of segregation. And again, the purple flowers were used here for the sorry purple flowers were used initially for the female part and white flowers for the male part. But now it is between the self, this was a self cross basically between the same flowers, so anyone can be male and anyone can be female, because the genetic makeup and the phenotypic makeup was similar for both the cases. So as I was telling you, this is an homozygous dominant, so it gave purple flower. This was a heterozygous dominant. It again gave the purple flower because purple was dominant over white. A heterozygous condition, again a purple flower, but in a homozygous recessive situation, we got a white flower. That was a one ratio. That means out of four, only one was that kind of a case. Other three were purple, and those were the dominant characters. Now to see whether the character was a pure purple or a heterozygous situation, as we were discussing earlier. So what he did is he crossed those F1 plants, those F2 plants, F2 flower plants with a white plant, just to see what would be the situation. If it were a completely homozygous dominant situation, then we must have gotten all of the purple flowers. And if it is a heterozygous situation, then we would get half of the offsprings as purple and half of the offsprings as white, 1:1 ratio. That is a short sort situation when there is a complete dominance and complete recessive kind of a situation that Mendel also got in his test cross, just to see whether the F2 generation was carrying a pure trait or it also has a white trait. But because purple being the dominant over white, only the purple flowers was there, and recessive flower was in the ratio of 1 only. That means 25% only. So for that, the test cross is the best method to know. Then he tested the two hypotheses for segregation in a dihybrid cross. What he did first was the hypothesis of sorry. What he did is he took two characters here: the yellow color and round shape, the green color and wrinkled shape. So yellow color and round shape was denoted by Y and capital Y and capital R. The green color and wrinkled shape was denoted by small y and small r. So the gametes, it gave us a capital Y, capital R in case of the yellow and round. In case of the green and wrinkled, it gives small y and small r. And the F1 generation, again, because it was a pure characters initially and then they were crossed, it gave us a heterozygous situation. But yellow color is dominant over green color, so it gave us yellow color only. And round shape is dominant over wrinkled shape, so the shape of the F1 generation was round only. Then after, when the F2 generation then after the F2 generation, when self crossed, then the experiment results contradicted the hypothesis. Now here the ratio was 9:3:3:1. So what did we got here is 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. It didn't came out to be 3:1. So he found out that the characters were actually getting assorted as independently, not with one another. He didn't found any kind of a linkage in the characters. So he found that the character yellow was not linked with round, and the green was not linked with the wrinkled. It was a complete independent situation. He found all four kind of combinations. He found yellow with wrinkled also. He found green with wrinkled also. He found green with round also. So the hypothesis of independent assortment was then devised here. So he gave so till now we have discussed two laws of Mendel. One is the law of segregation, second is the law of independent assortment. So basically this is just to show that segregation of alleles happen in ova, happen in sperm, and when the sperm and ova fuse to form the zygote, then it is totally very random event that which allele will fuse with which allele was present in the sperm and which allele was present in ova, that fuse to form the zygote, and then it will give us the combination accordingly. This is a totally chance event. Then came then another concept, that is the incomplete dominance, in which in this we saw that a single character, not a complete a single allele is completely dominant over other allele. It is not like that. Sorry, it is a incomplete dominance. That means that when a red color, a dominant character flower, was crossed with a white color, recessive color flower, so it should have given us all the red color flowers if it was a complete dominance situation. But no, it was not the case. The F1 generation, the flower that came out of after crossing this combination, we got is all the pink flowers. It gave us all pink flowers, not completely red. And when this F1 generation was self crossed, then it gave us three combinations, basically three type of flowers, basically a complete dominant red, a two incomplete dominant pink, and one white flower. Then again that challenged the Mendel's law of segregation, that how come this is possible, that a single sorry, Mendel's complete dominance theory, that how come it is possible that a single flower can give us uh, two pure color flowers can give us a mixture of colors. So then later it was found out that this was the complete concept of incomplete dominance, and this was studied in the snapdragon flower. So this was the actual flower. So after crossing we got this pink flower. Initially what was crossed was this red and this white, and we got after this crossing a pink flower. The concept of incomplete dominance. Then the multiple alleles for ABO blood group. So again here these two, A and B, are co-dominant alleles over O. So what happens is whenever A or AI, small I situation is present, then what is the phenotype that we will get? We will get a A blood group. When IB or IB small I situation is present, we'll get a B type of blood group. When AB situation is present, when IA or IB is present, then we'll get a co-dominant AB blood group, because these together can coexist and they together are co-dominant over O. But when we'll get small I situation, then only we will get a O blood group. So this is basically a multi alleles for a single character, that is blood group. So this is just to show, this is agglutination test that we do generally to check the blood group of any organism. So they have shown that here only. Another example is of polygenic inheritance, that is skin color. So skin color is actually controlled by three genes. Three genes means six alleles are the ones that control the expression of a skin color. So basically, depending upon which gene is involved in what proportion, the dominant alleles and the recessive alleles is what describes the skin color in a population. So it gives us a bell-shaped curve, because again, if you'll draw a Punnett square, so you'll get these kind of a combination. 1 out of 64 will be a completely dominant character. And then as the number of sorry, as the number of recessive alleles increases, the skin color also starts the skin color also starts getting darkened. So till the last situation where we'll get 1 out of 64, as all recessive alleles will have the uh, more skin complexion, darker complexion than the one in which all dominant color, all dominant genes were present. So this is basically, this is to show you how a polygenic inheritance of skin color occurs here. Again, three genes control the skin color expression. This is the effect of environment on the phenotype that is seen in these flowers. This again, then again, this is the pleiotropic effect of sickle cell allele in homozygotes. So two copies of sickle cell allele, all hemoglobin will be sickle cell type. It is an abnormal situation, it is not the normal situation. So abnormal hemoglobin crystallizes when oxygen content in blood is low, causing red blood cells to become sickle shaped. ठीक है. So whenever the oxygen content of blood is low, it causes the sickle shape of the RBCs, which can lead to many kind of diseases. So here a single change in a single allele or single gene can lead to its effect on many genes or many proteins, can affect the expression of many other genes. So it can lead to the breakdown of red blood cells, clumping of the cells and clogging of the small blood vessels, accumulation of sickle cells in spleen, which can lead to physical weakness, anemia, heart failure, brain hemorrhage, spleen damage, many other things which can lead to further many serious diseases. So here the change of the gene is in single gene, that is the shape of the RBCs. But it can lead to its effect on many other proteins, many other genes. So this is how this sickle cell works. ठीक है. So according to what is the chromosomal basis of inheritance that we have studied so far. So Mendelian inheritance has its physical basis in the behavior of chromosomes during sexual life cycle. Around 1900, cytologists and geneticists began to see parallel between the behavior of chromosomes and behavior of Mendelian factors. Chromosomes and genes are both present in pairs in diploid cell, as we have seen earlier, because as I was using the term allele. So basically, allele is the other factor, because the genes are always present in a diploid situation. It one When we are getting a chromosome, we have always 23 pairs of chromosome. That means every gene will have its pair on the its sister chromosome also. So that is how it is in a diploid situation, and it is the uh, gene will have its two alleles from both the parents. Homozygous chromosomes separate and alleles segregate during meiosis, and fertilization restores the paired condition of both the chromosome and genes. So basically, when the gametes are formed, when the female and male gametes are formed, then only the situation is haploid. After the fusion of these gametes to form the zygote, then all of the cells in the body will have a diploid condition, no haploid condition. Haploid condition is not a normal condition except when they are present in gametes. Okay. So this was the chromosomal basis of inheritance. The chromosomal basis of Mendel's law, again the fertilization among the F1 plants. So basically, yellow round seed and green wrinkled seed, they will undergo meiosis. Fertilization will happen, it will form gametes, and then the fertilization will happen. And what will happen during meiosis? The characters will be segregated. The characters will move to different poles to form the gametes. This you will understand if you guys have studied meiosis before. So meiosis is a kind of a cell division which actually helps in formation of the gametes. So characters will be segregated, it will lead to the formation of the gametes. These gametes will fuse to form the next generation, will fuse to form the zygote. So it shows us what the principle of segregation and principle of independent assortment. That means is that if you can see it here, the characters capital Y, character capital Y, character capital R, capital R, so they will segregate independent of each other. You won't see it here because these are the same characters, these are the same alleles on both the chromosomes. But if you'll come here, one has capital Y, one has small y. So it is not like the case if we have small y in one gamete, then it will have capital R only in its combination. No, it can have uh, sorry, it will not have small r in its combination always. It can have capital R also. So this is the law of independent assortment. And segregation means these all characters will be segregated into different gametes to form a complete set of chromosome, but a haploid set, not a diploid set. ठीक है. Then further, when this male and female gametes will fuse, it will complete this set to form a diploid set. And now the characters will come characters will come together and will give us the phenotype that was there in the genotype. So we can observe characters by phenotype only. Whatever is there in the genome will be shown, will be seen in the phenotype of that particular organism or plant. So it was the principle of segregation, principle of independent assortment. Now these both the chromosomes will be then separated to form further further kind of gametes for the next generation. Again, the independent assortment will happen. It can give us different kind of combinations. So again, independent assortment will happen and we'll have the next generation, which can lead to different kind of combinations, because these characters are independently assorted. It's not like they are linked to one another. Although the concept of linkage was later introduced also, in which it was shown that some characters are linked depending upon the distance upon which they are far from each other. Okay. So this was again the sex linked inheritance, in which if a character is present on the sex chromosomes of the parents, if it is present on the sex chromosome, then the inheritance will be sex linked, because females, in case of the Drosophila also, females have an XX type of sexual chromosomes, and males have XY. So obviously dosage composition is a different scenario, but the sex linked inheritance is a different scenario, because males will have a single X and females will have two X's. So depending upon that, also the inheritance of character is seen in case of Drosophila, or in all of those organisms in which this X-linked inheritance of characters happen, and there is a difference in the sex chromosomes that will determine either it will be a male or female. So this kind of inheritance is seen in that cases. Okay. So it is here for the eye color. Basically, red eye color is a dominant character and white eye color is the recessive character, wild type character and the recessive character. So it is an X-linked gene. Basically, in sex-linked inheritance also it is an X-linked gene. So what will happen is ova will produce all, because it is a homozygous dominant situation. So ova will produce all of the gametes in which they have a red eye character. Sperm will produce white eye character kind of gametes. When the fusion will happen, all of the offsprings will have a red eye because of the mother, because of the dominant situation in case of the mother. But when these the progeny from this F1 generation was crossed, then we got different combination. We got the females in which we got a red eye color. We got the female again in which we got the, all the females were red eye colored because of the mother, because mother had, or the X character that was passed on from father also had the no, not from the father. Father had the recessive character, but mother had the dominant character. And females have two X chromosomes, so it got compensated there and it got the red eye color. But the male here is what we got, white eyed male, because ova produced uh, two kind of sorry two kind of gametes. So we got one male that was white eyed, and all the females that were we got males in two combination, one with a white eye and one with the this red eye. And while in case of females, we got a combination in which we got homozygous dominant red eye and a heterozygous red eye kind of female. ठीक है, because the X that was coming from father also had the red eye character, and from the mother it has two combination, either a red eye, either a white eye. But again, as I was telling you, the females has two X chromosomes, and this is an X linked character, so it got compensated there. Although it was a heterozygous situation, still the female had the red eye. This is a dominant character. So this was a sex-linked inheritance because of the presence of character on X chromosome. So this was the cross then in which it was shown there ki how this happens. And this was an evidence of linked genes in Drosophila. So again, we are not going into those details. I hope you guys have understood it from the lecture. This is the meiosis I was telling you, recombination. How does this happens? So in meiosis, if you guys know about meiosis, this is a cell division, a type of cell division in which crossing over happens between the chromosomes, which can be a opposition of linked character. Basically, linked character are those characters which are present on the similar chromosome, and the characters which are very near to each other. So the characters which are very near to each other generally travel together. But if a crossing over kind of an event happens during meiosis, then those linked characters will show some kind of a recombination, and they won't be they won't go to a particular gamete together. They can be exchanged also just because of this crossing over event. So this is an example of showing recombination due to the crossing over. Ki how of Drosophila female having, sorry, how a Drosophila having this yes, female only having this gray with normal wings, and the male with, it is a double mutant. It has both the recessive characters, black with vestigial wings. And due to the crossing over, we got four type of combination: a wild type, a black vestigial type, a gray vestigial type, and black normal type. So this was because of the crossing over. Otherwise, these were the linked character. The combination, if the crossing over didn't happen, this wouldn't have happened. So this is the calculation of recombination frequency depending upon how much far a gene from a particular gene is, how much is the distance between two genes, is what tells us what would be the recombination frequency in that between those particular genes. Just a possibility of, again, due to the crossing over or due to the independent assortment, because of how they get arranged at the metaphasic plate in case of the meiosis. So this was again the life cycle of Caenorhabditis elegans, C. elegans basically. So depending upon at what our stage is the organism is, the different kind of events again happen in case of C. elegans. The sex determination in C. elegans is XX means hermaphrodites, XO means males. XO situation arises by a rare non-disjunction involving X chromosomes. Males can mate the hermaphrodites. Cross progeny are usually 50% males and 50% hermaphrodites. So this is a sex determination in case of the C. elegans. This is the lineage analysis in which, because the number of cells is less, so generally this lineage analysis can be done in case of C. elegans, which cell is forming what and what is getting formed from which cell, which mutation in which particular cell can mutate what. So this was a cell lineage study that was done on C. elegans. Lineage information is very useful to analyze the phenotype of mutants that can describe developmental defect with single cell resolution. Cell cell interaction, it can tell which cell is influencing what cell. Developmental potentials of a single cell can be told, and the time of commitment, at what time a cell is committed to form which organ, is also understood by the lineage studies. So this was what was there in the last week's lecture. I hope you guys understood it well. And now we'll take up the questions from the assignment. If anyone had any confusion till now, if anyone didn't understood any particular topic from the lectures, they can ask the questions. Otherwise, we'll move forward with the assignment questions. Any questions, anyone? हाँ जी, कोई questions किसी के? Otherwise, we'll move forward towards the assignment questions. I hope you guys have solved the assignment by yourselves. If not, and if you guys face some problem, then we'll discuss it here only. You guys can tell me your problems. No questions from lecture? Okay, guys, so if anyone is new in the class, let me just tell you how this works in this session. I'll speak the question and you guys will answer the question in chat box. Everyone will participate, everyone will write their answer in the chat box. No one will speak it loud so that everyone can perform and everyone can participate. Okay, so should we start with the assignment questions? Okay, then I'm starting with the assignment questions. So yes, so question number 1, the control of gene expression at the level of translation can occur by. Answer to question number 1, everyone. Answer to question number 1. I hope the questions are visible to you guys. So the question first, I am again repeating the question. The control of gene expression at the level of translation can occur by. This is the question and these are the options. ठीक है और Shalini, can there be more answers for this question? You can see the options, all of the 4 options, if you feel that any other option can be an answer. Other people don't want to answer. One and three, okay. So yes, in addition to the answer that you have already given, so the answer to this question is differential mRNA longevity, selective inhibition of mRNA translation, and ribosomal selectivity. ठीक है? So these 3 can be the answer. Alternative RNA splicing. The control of gene expression at the level of translation can occur by all these three: differential mRNA longevity, selective inhibition of mRNA translation, and ribosomal selectivity. Okay, next question. The binding of microRNAs at their associated RISCs to the 3' UTR can regulate translation by. Question number 2. The binding of microRNAs and their associated RISCs to the 3' UTR can regulate translation by. Yes, the answer is blocking translation initiation and recruitment of endonucleases to digest mRNA. Okay? Blocking translation initiation and recruitment of endonucleases to digest mRNA. Okay? Next question. Cis acting, 1 minute. Cis acting sequences that promote assembly of spliceosomes at RNA cleavage size are called. These are called what? Yes, it is splice enhancers. The answer is splice enhancers. Next question. Cis acting sequences that exclude exons from an mRNA sequence are called. Cis sequences that exclude exons from an mRNA sequence are called splicing enhancers. Right, it is splicing enhancers. Question number 5. A mutation in the blank of the myostatin gene lead to the formation of a larger muscle. Yes, splicing silencers. Question number 5. A mutation in the blank of the myostatin gene lead to the formation of larger muscles. What mutation in the myostatin gene? Question number 5. No, the answer to question number 5 is splice site. It is the splice site where mutation at which can lead to what? The formation of a larger muscles. ठीक है? Next question. Question number 6. Sickle cell anemia is a good example of which of the following? किसका अच्छा एग्जाम्पल है सिकल सेल एनीमिया? Question number 6. Yes, it is pleiotropy. Pleiotropy is the phenomenon जिसका sickle cell anemia is a good example. Next question. Why is C. elegans well suited for genetic experiments? Why is C. elegans well suited for genetic experiments? 2 and 4. The smaller genome size and shorter generation time. Good. One more answer to this question is the clonal propagation is easy. तभी तो उसकी lineage study हो पाई ना आराम से. ठीक है. Next question is, why does a frequency of recombination of 50%, what does a frequency of recombination of 50% indicate? Question number 8. What does a frequency of recombination of 50% indicate? Yes, the two genes are unlinked. Okay, the two genes are unlinked. Next question. In a genetic cross involving pea plants, flower of the plant chosen as female parent is covered to ensure that what? It is covered to ensure what? Question number 9. Mendel's experiment they are asking about. In a genetic cross involving pea plant's flower of the plant chosen as female parent is covered to ensure that question number 9 anyone. The answer to this question is it is protected from unintended pollination from any external source so that the results won't be affected. It won't pollinate from. Which option is not visible? Now can you see the option, Shalini? It is protected from unintended pollination from any external source. Okay, so this is the answer to this question. ठीक है जी. Next question is, sorry, in a dihybrid cross between a homozygous dominant and a homozygous recessive plant, phenotypic ratio of F2 generation. Now mind here, it is not about F1 generation, it is F2 generation is dash, which proves the hypothesis of dihybrid cross F2 generation. So answer to this question will be not F1, F2 they are asking about. Yes, it is. It would be 9:3:3:1, and it proves the hypothesis of independent assortment. Okay. Next question. MicroRNAs regulate gene expression by. They regulate gene expression by. Other options? Surely there can be more than one answers. 1, 2, and 3. All three options are its answers. Blocking translation initiation, recruiting enzymes that degrade mRNA, releasing the nascent polypeptide prematurely from the ribosomes. Okay. So with this, the assignment questions are over. I hope you guys didn't face any difficulty while doing so. If you guys didn't understood any question, we can discuss it here. Otherwise, I'll move towards the questions that I have brought for you guys. शुरू करें? Okay, perfect. So, first question, I hope it is visible to you guys. So, the first question is Mendel's law of segregation states what? Okay. Mendel's law of segregation states what? I am talking about law of segregation, not law of independent assortment. And it's not about the ratio. See the options on the screen. So, yes, the answer to this question is B. The two alleles of a gene separate during gamete formation. Okay? So the two alleles of a gene separates during gamete formation. Question number 2. The phenotypic ratio expected from a typical monohybrid cross between two heterozygotes showing complete dominance is. Very easy question. Complete dominance. I'm asking about phenotypic ratio, not genotypic ratio. So it should be 3 is to 1. Genotypic is 1 is to 2 is to 1, but phenotypic is 3 is to 1. Okay. So these are the things that matter in competition. The genotypic ratio from a cross between two heterozygote is. Now the answer. Yes, now the answer is 1 is to 2 is to 1. Okay, so now the answer is correct. Next question. In incomplete dominance, the heterozygote usually exhibits. Yes, the intermediate phenotype, because it is not completely dominant over other allele. Okay, codominance occurs when. Okay, codominance occurs when. Yes. Both alleles are expressed in the heterozygote. In the ABO blood group system, the IA and IB alleles are. मैंने बताया था आप लोगों को what is that? Codominance. Yes, it is codominant. A gene interaction in which one gene masks the expression of another gene at a different locus is called. Epistasis. Right, it is epistasis. In a recessive epistasis interaction, the phenotypic ratio in the F2 generation is commonly. Although आप लोगों को ये आज मैंने पढ़ाया नहीं है, but if you guys know the answer. Yes, it is 9 is to 3 is to 4. If someone doesn't knows that, then that's okay because ये आज आप लोगों को पढ़ाया नहीं है. Pleiotropy refers to the phenomena in which अभी हम लोगों ने डिस्कस किया है yes, one gene affects multiple phenotypic traits. ठीक है, one gene affects the multiple phenotypic traits. Now jump to question number 12, because again these two concepts we haven't discussed till now. So two genes are said to be linked when they, question number 12 we are talking about, when they 12 का BS are located close together on the same chromosome. ठीक है, when they are located on when they are located close together on the same chromosome. Okay, so the maximum recombination frequency between two genes is, what could be the maximum gene frequency recombination frequency between two genes is? No, it is 25%, because if 50% recombination frequency is there, then it won't be linked. ठीक है, so I think the question basically the question wanted to ask कि linkage के लिए what should be the maximum gene recombination frequency between two genes. Okay, so question number 14. One map unit or centimorgan corresponds approximately to what percentage of recombination frequency? If you guys know it, otherwise I'll tell you, it is 1% recombination frequency. So 1 map centimorgan is equals to 1% recombination frequency. ठीक है, crossing over occurs primarily during, if you guys have studied the cell cycle, in meiosis it happens, that's right, but in what phase of meiosis? If you guys don't know, I can tell you. 15th ka bhi, yes, it is Pectitin stage of prophase 1. It is Pectitin stage of prophase 1. Crossing over takes place between, this we have discussed today if I'm not wrong. Crossing over takes place between, yes, non-sister chromatids of homologous chromosome. It is between non-sister chromatids of homologous chromosomes. ठीक है, next question. In a 3-point test cross, the least frequency offsprings classes generally represent. This I'm not very sure if you guys will be knowing that. I'll tell you the answer. It is double crossovers. This is the phenomena you'll study further in the genetic series. Okay, a recombinant frequency of 12% between two genes indicates that genes are approximately how much far apart? We have discussed this just now. Question number 19, guys. Yes, 12 map units, because 1 centimorgan is equals to 1% of recombination frequency. So 12 map units or 12 centimorgan is equals to 12% of recombination frequency. Correct. Next question. A test cross is performed between. Question number 20. A test cross is performed between an individual of unknown genotype and a homozygous recessive individual, right? An individual of unknown genotype and a homozygous recessive individual. So with this we come to an end of the questions that I have brought for you guys. So now if any confusion is there, if you guys want to ask anything, then you can ask. I'm waiting for you guys. Otherwise you guys can leave the meeting. We're done with the questions. If you still haven't seen the videos, you want to ask them later, then we can discuss it next week also. There's no issue on that, but please do see the videos, guys. That will help you a lot. And if any one of you is preparing for CSIR or something, do pay attention towards the terminologies. It is very important while preparing for the exam. It's Gilbert. Gilbert is, if I'll say it in Hindi, it is a granth for developmental biology. उससे ऊपर कुछ भी नहीं है. Yeah, you're welcome. Any questions, anyone? Anyone wants to ask anything, they can. Otherwise, you guys can leave the meeting. Thank you for joining. We'll meet you next week, same time. Okay, no questions then. Bye, class. Thank you for joining.